Myself Sri Bhuvaneshwari, Department of Pharmaceutical Biotechnology. Today we will discuss about the isolation of microorganisms. So isolation of microorganisms from the pure culture. So to separate the microorganisms, so most of the organisms are generally found in nature like air, soil and water. So in the form of mixed culture. So here we can isolate the pure culture in a different methods. So here the diseased part of the plants and animals contain a great number of microorganisms which differ markedly from the microorganisms of other environment. To study the specific role played by the specific microorganism in its environment, one must isolate the same in pure culture. There are two steps involved in the isolation of pure culture. So here first, the culture has been diluted until the various individual microorganisms are separate apart from the agar surface. After incubations, they form a visible colonies. So after second, the isolated colonies has been aseptically picked off the isolated plate. So pure culture versus mixed culture. Pure cultures mainly originated from the single bacterial strains. So here the colonies are look like a same. Another mixed culture. So it is originated from the many bacterial strains. So this is different size and shape. We have to differentiate the species. So there are common method used for the isolation of pure culture. So there are so many steps involved in the isolation of pure culture. So the process of screening a pure culture by separating one type of microbes from the mixture is called isolation. So first method, the stick plate technique and port plate technique. So in port plate techniques there are two methods. One is loop dilution technique, another one is serial dilution techniques. Then spread plate technique, then micro manipulator method, then roll tube method. So in stick plate method, so in this method we are using the inoculating loop which is made up of tungsten wire. So this loop should be used for transferring the culture from the mixed culture to pure culture. So mainly the solid media is used for streaking the mixed culture in the agar medium. So there are the techniques used for the isolation of pure culture by stick plate technique. Here before streaking the mixed culture, we have to sterilize the inoculating loop with hot flame. So after red hot conditions, attaining the red hot conditions of the inoculating loop, after cooling a few minutes, then the mouth of the culture tube should be immediately sterilized. After that, the loop full of culture should be spreaded on the above the agar media, so like a zigzag manner by removing the loop backward and forward firmly. So after inoculating the mixed culture above the surface of the agar plate, agar medium, so it should be incubated overnight up to 37 degrees centigrade. After overnight incubation, the growth of the bacteria will be seen on the marked streaked marks. So next, pour plate technique. So here the liquid agar medium should be mixed thoroughly with the bacterial culture. After mixing the solid liquid media can poured in the sterilized petri plate and allowed for solidified and then incubated. So after incubation, the colonies appear on the surface of the agar. So these are the steps. So first, original bacterial culture is diluted with different culture tube. So each tube should be submerged or mixed with a fresh medium, then poured into the petri plate. So here after pore plate techniques. So there are some disadvantage in this technique. So the microorganisms are trapped beneath the surface of the medium when it is solidified. So here the surface of the colony should be identified is difficult because of the surface of the subsurface of the colonies. So this method also a tedious method and also time consuming and requires skill to identify the colonies. And next procedure for spread plate and pore plate technique. So this is a simple technique. So first we have to inoculate the sample in the surface of the agar plate, then spread over the surface with a spreader. So here after complete spreading above the surface of the agar, then we have to incubate at a 37 degrees centigrade by using the proper inoculation period. Then the colony should be identified on the surface of the agar. So same like pore plate technique. Here before introducing or pour the agar medium, so first we have to spread the mixed culture in the petri plate. Then you have to after pour the hot agar above the petri dish. After solidification, it should be incubated. After attaining incubation period, the colony should be produced. It's a serial dilution technique. So in this techniques, we have to successfully we have to choose the techniques which is not suitable for solid media. So here we can take a mixed culture isolated from the series of dilution. So we have to make a serial dilutions with a liquid medium. So here the number of tubes of sterile liquid mediums are inoculated with the liquids of each successive dilution. So here we can take sample. So each test tube we have to introduce 1 ml of the culture. So take 9 ml of fresh medium, introduce from the sample in each first test tube, that is 10 into minus 1, that is 1 by 10 dilutions. From this we have to introduce into 1 ml to the next test tube, that is 10 to the power minus 2 up to 10 to the power 6 or 10 up to the 10, because of the colonies, number of colonies will be reduced based on the dilutions. So after dilution, make serial dilutions, we have to again take loop full of organisms, that is spreaded same like the previous slides. We can discuss the agar spread on the agar plate or stick plate techniques. So up to the minimum number of colonies, we have to dilute all the serial dilution techniques. Spread plate techniques again on this one. Okay. So next spread plate techniques. So in this same bacterial suspension should be spreaded with the L rod. So after solidification of the solid agar medium, we have to inoculate. We have to spread over with the help of L rod. After incubations, we have to find out the number of colonies above the surface of the agar plate. So it is a simple techniques. This is method only for surface colonies are found. The microorganisms are not exposed to the high temperature. It's the advantage of spread plate techniques. So micro manipulator method. So in these techniques, see which permit one to pick out a single cell from a mixed culture. So here the instrument which is used to pick out the single colonies by this method. So this is the advantage, but can be reasonably show that the culture come from single cell and one can obtain the strain within the species. So it is a equipment is expensive and it is manipulation is very tedious and it is required as skill for operation. So it's the techniques how to pick up the colonies with the help of the instrument micro manipulator. Thank you.